Hi everyone, so in this video I'll talk about real-time PCR. So let's get started. So what is real-time PCR? So in today's world, quantification is carried out by real-time PCR, which is a modification of the standard PCR technique in which synthesis of the product is measured over time as the PCR proceeds through its series of cycles. So real-time PCR is very simple, or RT-PCR we call it, as it will detect or it will calculate the amount of amplicons or product produced over time throughout the entire PCR reaction. So there are two ways to follow or to calculate the product synthesis in real time. So one is the use of fluorescent dye that gives fluorescent signals when it binds to the DNA, and other is the reporter dye, which is a short oligonucleotide which gives a fluorescent signal as well when it hybridizes to the PCR product. So these are the two methods in which we can keep a hold of the products or the synthesized product over time. So moving with that, so talking about the first method, which is the dye method, which we'll be talking about. So a dye which can be used is a ethidium bromide. So let's say ethidium bromide is a good option to be used as a dye in these RT-PCR. So the use of ethidium bromide is to quantify PCR products as they accumulate. Also, amplification produces increasing amounts of double-stranded DNA which binds ethidium bromide, resulting in increasing fluorescence. So as they are increasing amplicons, so after a PCR cycle or after numerous cycles, as the number of products or the amount of product increases, the fluorescent signals will also increase, which will directly be proportional to the amount of fluorescence produced over time. So an alternative to ethidium bromide is SYBR green one, which can be also used in place of ethidium bromide. So this particular dye binds in the minor group of double-stranded DNA, and on binding it produces increases over 100 folds. So problem of these two dyes is it might bind to primers or other non-specific areas which we don't need other than the target DNA. So these are the problems that are related with these dyes. So moving with that, so talking about the reporter probe, which is a very important topic which I want to cover in this video, so which I'll be explaining you thoroughly how it happens. So the most widely used probe is TACMAN. So reporter probe used is TACMAN. So reporter probe is a very unique thing that is used in RT-PCR and used at a very unique point of time, and it has produced and it has resulted in highest accuracy that is produced. So in this system, the probe used are oligonucleotides with reporter fluorescent dye attached at the 5 end and a quencher dye at the 3 end. So you can see the first diagram here. So in between, this is the probe, you see, right? You see the purple patch, which is the probe, and we have two dyes here, or this is the reporter dye at the 5 end and this is the quencher dye at the 3 end. All right, so moving to the second one, which is while the probe is intact, the proximity of the quencher reduces the fluorescence emitted by the reporter dye. So what happens in this case? So coming to the second diagram, which is, so you can see when these two dyes are in close proximity or are together attached to the probe, what quencher dye does that it reduces the fluorescent signals of reporter dye. So it will result in less fluorescence produced over time when the reporter dye is attached to the quencher dye. So due to the presence of quencher dye and reporter dye together, this results in decrease in the fluorescent signals of the reporter dye. So coming to the third point, which is if the target sequence is present, the probe anneals downstream from one of the primer sites. So if the probe finds a target site, so it will bind to that target downstream from one of the primer sites, which can be forward or reverse primer. So as the primer is extended, the probe is cleaved by the 5 nuclease activity of the Taq polymerase. So you can come to the third diagram, which says that as the PCR cycle proceeds, so these primers which are used, which is the forward and the reverse primer, will also proceed throughout the cycle. So as these primers proceed in the respective directions, it will result in cleaving of the probe. So as they move forward, so as the 5 end and the 3 end primers move forward, this results in cleaving of the reporter dye. So as this particular dye move forward, this results in cleaving of the reporter dye from the probe. So now in this case, So now you see the next diagram, the reporter dye is free and independent now, whereas now the probe is only attached to the quencher dye. Alright, so this happens in this case. So come to the next point, which is this cleavage of the probe separates the reporter dye from the quencher dye, thereby increasing the reporter dye signal. So now that the reporter dye is independent and is not attached to the quencher dye, now it regains its fluorescent signals and produces good amount of fluorescence, which was not the case when it was attached to the quencher dye, because quencher dye being in close proximity with the reporter dye reduces the fluorescent signals of the reporter dye. So now that the reporter dye is independent, it will regain its fluorescent signals as well. Also, so come to the next point, which is cleavage removes the probe from the target strand, allowing primer extension to continue to the end of the template strand. So as this thing goes on, the primers will keep on moving and move throughout the cycle. So come to the last diagram, as you can see here. So as you can see, the 5 end and the 3 end primer have traveled across throughout. All right, so these will keep on traveling as long as the PCR continues, and these will result in cleaving of more of these reporter dyes. So there are more of these reporter dyes. So there is not just one reporter dye present in the whole PCR reaction. There are numerous reporter and quencher dyes present in the entire PCR reaction. So as the PCR goes on for many cycles, let's say 25 or 30 cycles, so it will keep on cleaving the reporter dye in every cycle. So this is just a pictorial view of just one cycle. So this happens the same way in all other cycles. So it will keep on cleaving the reporter dye in every cycle, and thus the amount of reporter dye or the population of reporter dye would increase drastically, and thus the fluorescent signal would also increase. Alright, so as the fluorescent signals increase, it will lead to, or it indicates that the amount of amplicons produced are directly proportional to the fluorescent signals as well. So as the fluorescent signals or the fluorescence increases with time, the amount of amplicons increases as well. So this is the method, or this is the idea of using a reporter dye. Alright, so this is done, and additional reporter dye molecules are cleaved from the respective probes with each cycle, affecting increase in fluorescence intensity to the amount of amplicons produced. So that is just what I said. So as they keep on traveling through the PCR cycle, the amount of reporter dyes increase over time, which will indicate the amount of amplicons produced over time and increase in fluorescent signals as well. So coming to the last point, which says that quantification of the amount of target is unknown in unknown samples is achieved by preparing a standard curve. So if you want to know, or if in case we want to know the concentration of or quantification or amount of product produced in an unknown sample, we can we can plot a graph, and with the help of standard curve we can determine the concentration of the unknown samples as well. So moving with that, so coming to the next part, which is a molecular beacon probes, so which is an alternative to reporter probes. So this is an alternative that is used to reporter probes, which is a molecular beacon probes. So these differ from the Tachman probes in that fluorescence occurs by hybridization of the probe. So in this case, this occurs by hybridization of the probe rather than cleavage of the probe during amplification. So in Tachman probe, what we observe that it occurs through amplification or cleavage of the probe. So this particular probe, which contains a reporter and quencher, so in Tachman what happens is it will lead to removal of the reporter dye, and thereby the increase in fluorescent signals takes place. So this is the logic for Tachman. But whereas in molecular beacon probes, it occurs by hybridization of the probe. Alright, so this is, you can see, this is the probe entirely. So this is the oligonucleotide probe, which contains quencher compound, which contains the fluorescent label. Along with that, we have the DNA target. So when the DNA target hybridizes with the probe, it produces a fluorescent signal. Here you can see. So when both of these are together, it will produce a fluorescent signal. So molecular beacons are hairpin shaped oligonucleotides with a fluorophore at one end and a fluorescent quencher at one other end. So the structure is a bit different on this case. So we have a fluorophore at one end and a fluorescent quencher at the other end. So while in the hairpin, structure of the fluorescence is quenched, but when the probe binds to the target, it undergoes a conformational reorganization that restores fluorescence. So definitely when hybridization occurs, it will result in formation of more of the fluorescent signals. So moving with that, so I'm talking about the real-time PCR applications, which is the last part of this video, which is some of the real-time PCR is often used to quantify the amount of DNA in an extract. So for example, to follow the progression of a viral infection by measuring the amount of pathogen DNA that is produced in the uh, present in a tissue. So it is very useful for finding a solution to any disease or any pathogen, or exploring a pathogen that is newly occurred. So real-time PCR is very useful for checking or for quantifying the amount of DNA in a particular extract. Also measuring the RNA amounts in particular to determine the extent of repression of a particular gene by quantifying its mRNA. So the gene under study might be that is switched on in cancerous cells, and in which case quantifying its mRNA will enable the development of cancer to be monitored and effects of subsequent treatment can be accessed. So real-time PCR has numerous applications like this for checking, for studying the cancer cells as well, using the real-time application of PCR. So let's just keep this video till here. Hope you enjoyed this video, and thanks for watching.